Good morning, students. Very good morning, everyone. Good morning. Happy morning to all of you. And so, directly come to the topic today. See, before Independence Day, we'll be discussing some common topics in geology. Okay, so that we develop a perspective and also realize the importance of something called PYQ, previous year questions. Okay, after completion of every chapter, make sure, and in fact, I will also make sure and I will make you do, even if you don't have time to do any other exercise, but the previous year questions should not be left. In fact, after January, the PYQs have to be revised again. And all those questions where you did not find the answer or you knew the answer, but somehow you make a mistake. You make a mistake maybe due to overseeing it. Okay. So in that case also, revision of these questions after January, I mean from January onwards. PYQ is the key here. Always remember. Okay. In fact, let me tell you, in the North India, doing PYQs is a part of track sheet there. It's a part of track sheet after January because it is important. Now, let me take today the paper of the year 2021. Okay. We have seen in the previous class the paper of 2022 and we have done some questions. Now I'll take 2021 and if time permits or if your doubts are there, I will again go to 2022 and I'll take questions from that also. So here goes paper of 2021. Now, the distribution of zoology and botany is a little erratic in Andhra Pradesh. Or I should say it is not same as in rest of India or maybe in the central or north India. In north and central India, topics like ecology is in botany, topics like genetics. Am I audible also? I will start the chapter after the Independence Day. We will start the track sheet from that day. Before that, every year we do this type of treatment wherein we tell the student that whatever the questions that you have written this year are actually from the knowledge that you have already gained or can easily gain. There is no rocket science in those questions. A little confidence and a smart revision. It is not that all the selected students, all those who are on the merit list, they will be able to complete whatever they decided. I have not seen anyone who said that, sir, I had a plan this much, and by the end of the session, yes, my plan is over. Always people have higher hopes, higher targets, and some or the other thing is not done. So do not worry about that. It is everyone is facing the same thing. But whatever you know, doing the best in that confidence is to be there, and we will develop this confidence by doing these type of bits. Hope are you there with me? Okay. So let me take you to paper of 2021. Let's go. Now, the botany portion, some questions are of zoology, but they are in the some questions of botany are in the zoology syllabus of Andhra. Since I am here in Andhra for the last 6 years, 5+ years, so I am also taking them in zoology only. Let us do this. Which of the following statements is not correct? Find out the incorrect one. And this is about the pyramids, okay? The ecological pyramids or Eltonian pyramids. We also call them Eltonian pyramids, okay? So three type of pyramids are known to us, right? Three type of pyramids are known to us. Let us recall how many of you remember. Let me check the ecological pyramids. ecological pyramids. What are those three types? Okay. One is pyramid of number, pyramid of number. One is pyramid of biomass and pyramid of energy. As we know that energy keeps on decreasing as we are moving higher the trophic level, energy to pakka decrease. So T1 energy at T2 is lesser than T1 and subsequently energy is decreasing. And therefore, like this size shows the amount of energy, this size lesser means lesser amount of energy, like that. So we will get an upright type of pyramid. Now you may be asking, sir, pyramids are always upright. Yes. But here, when we are trying to make, sometimes we also get the inverted pyramids, and that is the place where questions come. 99% questions come on the inverted pyramid. So let us see what are those pyramids which are inverted. Inverted pyramids, inverted ones, inverted ones. In case of pyramid of energy, there is nil, no as such, okay? No pyramid which is inverted. Pyramid of biomass, who will tell me which pyramid is inverted in the pyramid of biomass? Beta, who will tell me which pyramid is? Yes, you're right. Aquatic ecosystem. So we are talking about the inverted one, and in the aquatic ecosystems, we have an inverted pyramid. We have an inverted pyramids. Pyramid will be drawn like this. Now, what does this mean? This means that the total biomass of the lower trophic level is lesser than that of the higher trophic level. Now, this may look paradoxical, but let me first write down what this means. T1, T2, T3, and T4, trophic level 4. If it is a ponds ecosystem, what is T1? T1 is phytoplanktons. T2 is zooplanktons. T3 are sometimes the small fish or sometimes mollusca. I'm writing small fish. And T4 that represents the larger fish. Okay. That is smaller fish, and T4 is larger fish. What has been noticed that the biomass, the total organic matter, keeps on decreasing. The total organic matter keeps on decreasing. This is T1, T2 is zooplanktons, T3 is small fish, and T4 is the larger fish. The total biomass of the zooplanktons is found to be more than the total biomass of the phytoplanktons. Similarly, the total biomass of the zooplanktons is lesser than that of the small fish. And now you understand the trend. What is the trend here? You have understood, okay? Which means that a smaller standing crop is sustaining a larger standing crop. Now, how it is possible? Suppose if in my whole life, I am only made available 10 kilogram of food, how come my weight can become 15 kilogram? It is certainly much lesser than 10 kilogram. It cannot be more than 10 kilogram. If in my whole life span, I am being given 10 kilogram of food. So this looks paradoxical. Normally it doesn't happen. Normally what we find that energy and biomass, even we also use them in terms of the finding out the uh, population density also. We can represent population density by biomass also. So biomass and see, it is almost like comparable. Energy, biomass gives energy. How come a lesser biomass can give comparatively higher energy? No, it is not possible. Anyone who can answer why this paradox in aquatic ecosystem? How this is possible? A smaller standing crop can sustain a larger standing crop. If I am only given 10 kilogram of food, my weight cannot be more than 10 kilogram. So how can you, yeah, I mean, in my whole generation, number is also inverted. Number is also inverted. Mahima, you are right, beta, but we will talk to pyramid of number. Right now, we are discussing the pyramid of biomass. Biomass means total organic matter. Total organic matter. Suppose if you collect all the zooplanktons and all the phytoplanktons, you take the dry weight. So that dry weight represents the biomass, the dry weight. You can also do it in fresh weight, but it is very good to do it in dry weight. So the dry weight of all the phytoplanktons is called the standing crop at T1. What it's called? Standing crop at T1. Make sense? Similarly, the total biomass of all the zooplanktons can be called the standing crop at T2. I hope if you recall the definition of standing crop, you can easily remember it is the total biomass at any trophic level. Total organic matter, biomass ante organic matter. Okay. Now, one student number is also inverted, 10% loss, sir. Ha, beta, to 10% law is not applicable. Na? In case of 10% law, the biomass should decrease. Because if the energy is decreasing, certainly the biomass available is also lesser. Okay, then only energy can be lesser. If I eat 10 mangoes and I'll be having some energy, I'll gain some energy. If I eat 20 mangoes, I will gain more energy. So here biomass can be related to the energy. You are right. As pyramid of energy is upright, how come this pyramid can be inverted? Any one also, if you can try, then I will come to my answer. Any one of you want to try it, I'll then come to my answer. No one? This answer is given by Taju. Mahima has given my Priyanka. Aquatic, yes, you answered correctly. Single tree ecosystem is pyramid of number is inverted. Yes, beta. We'll come to pyramid of number. This is pyramid of number. We'll discuss, right now we are discussing the pyramid of biomass. We are only discussing the inverted pyramids. Okay, your answer is correct. We'll discuss about that. Okay. Okay, I'll come to this answer. Before that, I will complete this flow chart as some people are asking. The pyramid of number in a tree ecosystem is inverted. In a tree ecosystem, generally we take one tree. Okay, so one tree has got, let us say, 200 small birds. 200 small birds have got the parasitic insect. Every small bird has got 10,000 parasitic insects like that. Okay, so we get this type of pyramid. Sometimes, in case of the pyramid of number in the tree ecosystem, we also get this type of pyramid. So far it is same, but then we also get this. You understand? Which is a spindle shape. It is a different type of inverted and it is called spindle shaped pyramid. In the same tree ecosystem, we can even get a spindle shape. So both the shapes are applicable in the single tree ecosystem. Tree ecosystem. Either they will give the option as tree. If they give forest ecosystem, then also your answer should be same. A forest ecosystem can also be equated in terms of single tree. In the tree ecosystem, pyramid of number, because the tree, no matter how much big is the tree, how much large is the tree, how much heavy is the tree, in terms of number, it is a single tree. So maybe the biomass follows the 10% law, but the number is inverted. Okay? I'll tell you how this also comes. I'll tell you how this comes. I will tell you, but before that, I hope you have remembered once again. Let us remember with me. The inverted pyramids, in case of pyramid of number, the tree ecosystem, inverted pyramid in pyramid of biomass is aquatic ecosystem, and pyramid of energy is never inverted. Are you guys with me? And now I'm giving the answer to this question. How it is possible? See, because the lifespan of these different organisms is different. I mean to say, the lifespan, the total lifespan of the phytoplanktons is much smaller than that of the zooplanktons. A zooplankton takes more time to complete one life cycle. In that duration, 2-3 generations of phytoplanktons are completed. In that duration, 2-3. So what happens that this one generation is actually dependent on, or it has fed on more than one generation of the lower trophic level because of differences in the duration of the lifespan. Because of differences in the duration of the lifespan. Make sense? Okay. So that is the reason. Similarly, as you go higher the trophic level, the lifespan is increasing. Okay. So the small fish, they have got a longer lifespan than zooplanktons, and the larger fish has even a longer lifespan or life cycle. And that is the reason, rather than one generation, they feed on multiple generations of the lower trophic level. Am I clear? Is this point clear to everyone? Multiple generations of the lower trophic level. Is this point clear to everyone? Good. Okay. Now, lastly, I have to tell you how we get the spindle pyramid. In the NCERT, if you read the page where pyramids are explained, they have given one small exercise to the student. They say that, let us say there is a big banyan tree. On that banyan tree, there are 200 birds. And those birds are eaten up by larger birds. Smaller birds are eaten up by the larger birds. Okay. So how many banyan tree? Okati. How many small birds? 200. How many larger birds? 35. I'm just giving an example. So this is a spindle shaped pyramid. It is a modification of inverted only, because how I drew it like this, beta. I first make an inverted one and then I make it a spindle shape. You understand? It's called spindle shaped. Spindle shaped. Spindle shaped can be obtained in the tree ecosystem. This is possible in the tree ecosystem with the example I have given you, and this is important. This is important. If you read the NCERT, you will find example of this ecosystem and this pyramid. They will ask the student only, what type of pyramid will you obtain? And the answer is, we are going to obtain a spindle-shaped pyramid. My dear children, hope the pyramid's point is now revised to all of you. And any such question based on the pyramids, I hope you will be able to answer. Provided that only, even if you are listening to me 85 to 90%, then also you will be able to answer the questions. With every question, you are becoming more and more immune. You are becoming more and more immune to mistakes. Dear children, in the equation GPP minus R, what R represents? R represents what R represents? R represents respiratory losses. Easy question. I'm not discussing it too much. The amount of nutrients such as carbon, nitrogen, phosphorus, and calcium present in the soil at any given time is referred to as, come on, the amount of inorganic nutrients. Many insects, yes, that also correct. One in yes, single tree, many insect and less number of small birds, and very few large birds. Very good, very good. Yes, you are right. Standing state. So Mahima and one more student have answered, and that is the correct standing state. So please understand there are two terms. One is standing crop and one is standing state. Now, crop represents the organic matter. Crop represents the organic matter, the total biomass, okay, the total biomass at a given trophic level. That is standing crop. whereas standing state is the total inorganic nutrients. And where these nutrients are present, of course, they are present in the soil. The total inorganic nutrients present in the soil at any given point of time is called standing state. Now, if you are confused between crop and state, you can relate crop. Crop means plants, crop means life, life is organic, like that you can relate, okay? And state is other, so you can easily understand what is crop, and that's how you can differentiate them. When gene targeting involving gene amplification is attempted in an individual's tissue to treat disease, it is known as, okay? So gene therapy, different types of gene therapies are being done. One type of gene therapy is totally replacing the affected gene, okay? Sometimes gene amplification of the target gene is also done. It is a type of gene therapy. Once again, now I hope you are heard it, and now listen to the statement. When gene targeting involving gene amplification is attempted in an individual tissue to treat disease. So when we are using the genes to treat a disease, it is always gene therapy. It is always gene therapy. We are treating the disease, so it cannot be molecular diagnosis, because without the diagnosis, therapy or treatment won't start. So treatment will only start after the diagnosis, okay? Please remember that. Gene therapy is a collection of methods that allows correction of gene defect that has been diagnosed in a child or in an embryo. Biopiracy is the term used to refer to the use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned, without compensatory payment. So if a company in America is using the neem as a director, and they are making a type of toothpaste out of neem, and they are minting money, they are becoming millionaires. And this is based on our knowledge of Ayurveda, our local knowledge. In India, the local knowledge, we know that neem is good for the teeth. Neem is good for multiple things, but if these companies are minting money, they are minting profits because they are too big firms. No one can, say to them directly, and therefore there are organizations which help the countries or the people against this crime. And what is this crime known as? It is called piracy. But it is a piracy. Piracy means cheating, duplicate. Piracy of the biological matter. So it's called biopiracy. In the same way, if any biological matter is used to affect a population or kill the target population, if any biological knowledge is used, that is called bioterrorism. Bioterrorism. If uh, someone's knowledge is used to make big profits, make a business out of it without giving the compensation. If you give the compensation to the Indian government, it is okay. You have taken the license, you have taken the license to make neem oil or neem toothpaste in your country. But without compensation, without the license, license means compensation. License will come only after compensation. So you understand biopiracy, and molecular diagnosis refers to the act or process of determining the nature of cause of a disease. Nature and cause of a disease. That is called diagnosis. That is called diagnosis. Okay, simple. Let us go to the next one. In spite of interspecific competition in nature, which mechanism the competing species might have evolved for their survival? And here the answer is how the competing species avoid competition. If you remember MacArthur, a naturalist, MacArthur observed that different species of warblers, he observed 5 different species of tree warblers. Yes, very good. Birds, food timing, very good. He has studied 5 different species of tree warblers. Tree warblers are a small birds which are feeding on trees, fruits. He found that although their food is same, their food is seeds, that is same, still they are not competing with each other and none of them are harmed because in competition it is a minus minus type of interaction. This minus minus is not seen. So how do they overcome competition? Answer is they overcome competition by a phenomenon called coexistence. Coexistence. A phenomenon called coexistence. Now, how come coexistence is possible? How it is possible? Uh, how it is possible? MacArthur said it is possible by something known as resource partitioning. He said resource partitioning. What is the resource? The seeds. Resource partitioning. Partitioning. Now, how you can partition your resource? There are a variety of ways. If the resource is the food, you can partition the resource by having different foraging behavior. Different foraging behavior. Foraging behavior is all those behavior of an animal to obtain the food. An animal adopt all the mechanisms to obtain the food. See, foraging means obtaining the food, capturing the food. Foraging behavior. Foraging behavior, that is not same, that is different. This is how they do resource partitioning. Now, an example of different foraging behavior is some birds, they only forage in the apical part of the tree. They fly to the top and they only feed on the places at the top. Some do it in the bottom, some do it in the middle part. Even in the middle part, some do it in the periphery, some go to the core. Even if two species are doing at the same place, let us say two species are doing at the bottom of the tree, they may have different timing of foraging. For example, one can forage in the early hours of the day, the other one can forage in the middle part of the day. So they may have different foraging behavior and different foraging timing. Different foraging behavior, different foraging timing. Make sense? Understood? Everyone got it? Any issue so far? Standing state. Sir, we could see answers. Okay. Birds. Okay. Beta, Vivo 1906. Beta, actually, we are not doing any test right now. So I know that answers are underlined, but actually, I am discussing the paper. So you can, and also I'm not only discussing the answer, I'm discussing the allied points also. So basically, we are learning some matter with the help of MCQ. This is called MCQ feedback. We are taking the feedback from the questions. Whatever learning we are doing through the questions. Okay? This is known as MCQ feedback. Okay? However, I will try next time to bring the file without the answers, but even right now also you can utilize it. Another question, question number 135, it says immensialism can be represented as. Now, immensialism, you know what is that? Who will answer immensialism? Who will answer? Come on. It is minus and zero type of interaction. Very good. Minus zero type of interaction. Okay. Any example of that? You know, a fungus called Penicillium notatum. You know, a fungus called Penicillium notatum. Now, Penicillium notatum secretes a chemical, and due to that chemical, the bacterial growth is affected. Bacteria could not grow around that area. Wherever the penicillium is growing, bacteria cannot grow in that area. Why? It is found that some chemical released from the fungus kill the bacteria. Okay. What that chemical? It is a toxin to bacteria. Okay. What is the advantage to the penicillium? No advantage. There is zero. Penicillium is at zero. Bacteria, they are killed, they are minus. So, all examples of antibiosis, where we obtain antibiotics from the natural environment, we find that it is all of them are examples of amensalism. They are example of amensalism. Am I clear to you? You understood? Amensalism. Amensalism means there is no apparent advantage to one species, but other species is harmed due to the presence of the first species. Now, DNA fingerprinting involves identifying differences in some specific region in DNA sequence called as. Now, you have to find out what is the answer. Now, differences in some specific region of the DNA. Some specific region. What is that region? That region is repetitive DNA. That region is repetitive DNA. In the repetition, the number of repeats are not same because of mutations, because of accumulation of mutations in these parts. This is a straight line taken from the NCERT. The statement is exactly NCERT line. I think the first paragraph of DNA fingerprinting, you will find this line, exact line. Another one, in the exponential growth equation, NT is equal to N naught e to the power of RT. What is NT? NT means population density after time T. Okay. Banyan tree and small herbs. Banyan tree and small herbs. Okay, I'm not able to recall this banyan tree and small herbs, how they are immensialism. Anyways, I am now explaining you this term. Okay, so NT is the population density after time T, and not or N0 is population density, current density right now. Then E, what is E? E is the base of natural logarithm. It is the base of natural logarithm. What is R? R means, anyone? What is R? Who will tell me what is R? How you obtain R? What is the, what two things are the difference of the two things? What is that? Come on. R. Yes, R can be R can be called the per capita growth rate or intrinsic rate of natural increase. Intrinsic rate of natural increase. Per capita growth rate. What is per capita birth rate? Total birth upon the population density. Per capita death rate, total death upon the population density. Take the difference of the two, it is the per capita growth rate, or R. Birth rate minus death rate is equal to R. And R can be called per capita growth rate, or what else? IRNI, intrinsic rate of natural increase. After R, what is the other thing here? T, T for time, the duration, the time, that is T. Simple one. They will not give you any question on, solving the, questions. Yes, they may ask you what are these different symbols in an equation mean. Nowadays, it is possible to detect the mutated gene causing cancer by allowing radioactive probe to hybridize its complementary DNA in a clone of cell, followed by its detection using autoradiography. Because how you can detect any mutation in the clone of cell? First of all, what is a clone of cell? Let us understand all this. Why we are doing this? Suppose the doctor has identified that there is a lump. There is a lump in the neck. There is a mass in the neck, and we want to test whether some cancer is there or not. We want to test some cancer is there or not. Okay. What we can do? We can put a long needle inside and take some tissue out of it. We take a very little amount of the tissue and we collect. So this is a sample obtained. Sample. Now we have to multiply the sample because you have to be sure about your answer. It is better to multiply the sample. Okay. So for that we can obtain a clone of these cells. Here few cells are there, and now this is a clone. That means a large population of cells. In the clone, now we are testing its DNA. Now DNA is double-stranded, as we know. There is a complementary base pairs are there. Complementary base pairs A, T, C, G. Complementary base pairs are there. Now we have to check whether these base pairs are same as normal or they are some different. Normal already we know. Normal already we know. So what is normal? One of these strand which is complementary to the other strand, we will prepare in the laboratory. And what is the preparation required here? Then we will label it with radioisotope. It is labeled with radioisotope. It is a single-stranded DNA labeled by radioisotope containing the normal sequence, normal complementary sequence. It is a normal complementary sequence, okay? Radio labeled. This is a radio labeled DNA probe. Radio labeled DNA probe. Understand? Single stranded DNA. Now you have to try to hybridize this with our cells, the DNA inside the cell. For that, you have to denature it. Okay. So by using some denaturation agent, we have to denature these. So after denaturation, the two strands will be separated. The two strands will be separated. Okay. So by some technique, we try to leave only one strand, and this is the main strand against which the probe has been prepared. So if there is A here, there is T in the normal one. If there is C here, there is G in the normal one. Okay. So we have taken this complementary to the one which is now left in the sample. Now what is to be done? Simple, hybridize them. Wait for a while. Wait for a while. This is a DNA in the sample. This is a radio labeled probe, and this is after hybridization. Okay. Now you have to check it whether you are finding any radiations, any such wavelengths from the sample, so that how you can do? You can use some frequency scanners, or very simple, very cheapest method is use some photographic plate or simply X-ray plate. You can use a photographic plate, and we keep, suppose the sample is in this plate, and under that we keep the photographic plate and leave it in the darkness for some time, and then we check the photographic plate. If in the photographic plate we find some scintillations, some lines are there, it means that why these lines will be there? The line will be there because the radiation coming out from the sample. And why the radiations are coming out from the sample? It may come out because there is a radio labeled probe hybridized with its complementary one. Very, very simple. So if you find any line, any mark, it indicates that in the sample which you are testing now, the radio labeled probe is present, and it will only present because the probe is complementary to the normal sequence. So if it is present, it means that the natural DNA in the cell is having the normal sequence. Let us take an example. If in place of a particular base pair, let us say in place of G, a T has come here. a single base pair. Suppose if there is, what will happen now? Because at that locus in the DNA probe, okay, so in place of T, in the DNA probe, G was there, and here it is T, and they are G and T are not complementary. What will happen? Eventually, this probe will not hybridize with our sample DNA. As a result of that, when we wash it multiple times, even the probe, DNA probe will be washed out. Why it is washed out? Because it is not complementary. No complementarity. Therefore, it is washed out. And when it is washed out, okay, when you take a scan or when you take the photographic plate scan, you will not see anything. You will see darkness. A darkness indicates that the probe has not hybridized. A probe will not hybridize only because if there is some mutations in the DNA. So darkness represents mutation. A bright mark represents normal gene. Okay? You understand? Darkness represents mutation. If a mark is there, it means normal gene is there. You understand? This line is important. If a mark comes, it means normal. If a negative report comes, negative means no mark is there. It is really negative. That means it indicates cancer. Okay. I have explained you. Now let me just check whether you have really understood or not. You have 4 options in front of you. Let us answer them. Okay, let us see who will answer them. Let us answer these. 4 options are there. This question, nowadays it is possible. You can read all the 4 options. You can read all the 4 options. Answer cannot be seen. Let us see what is the correct answer. Who will tell me the correct answer? Remember that this Gosalites Zoology 4, it is a different thing. It is not the answer, beta. It is a different thing. Do not consider it as an answer. Come on. Yes, 1, 2, 3, 4. What is the answer? Once again, let me ask you. If you don't find any mark, what does that indicate? Normal or abnormal? If you don't see any mark, will that indicate mutation or no mutation? Normal. Mutation, leather normal. Yes, no mark. Come on. If there is no mark, it is abnormal. It is mutations. Very good. Once again, if you are not able to think, you can just think. If there is no mark in the plate, in the photographic paper, it means that the DNA probe has not hybridized. And why the DNA probe cannot hybridize? If the sample DNA, which is present in the real sample, if it is not complementary. And why it is not complementary? Because we have made it complementary in the laboratory. It is not complementary because, or might be that some sequence has been altered. And what is this alteration in the sequence? It is mutation. So because of mutation, there is no. Now let us see. 1, 2, 3, 4. What is the answer? Check the answer. Mutated gene does not appear on photographic film as the probe has complementarity with it. As the probe has complementarity with it. Complementarity. Option number 2, mutated gene partially appears on a photographic film. Option number 3, mutated gene completely and clearly appears on a photographic film. And option number D, mutated gene does not appear on a photographic film as the probe has no complementarity with it. Come on, answer. One student has answered, Hema has answered, first Hema answered 1, then she answered 4. Well, I'm not sure what is your answer. Priyanka has answered. Let us see who others can answer. I want at least 5 more answers. Just now I have explained this, okay? And Deepika has given a wrong answer. V2353, 2153. No, it's not correct answer. Mohit has also not given the correct answer. Shreya Raj, no, Mohit has given the correct answer. Mohit, Shreya, they have given the correct answer. Hema, sorry beta, your answer is wrong now. Lenovo K6 Note, your answer is correct. Okay, so let us, okay, your Shweta. Shweta, your answer is correct. Yes, if you are giving the answer from yourself, that means you are really understanding. 1, 2, 3, your answer is beta. Okay, okay, you will decide what is right, what is wrong. Mutated gene, that means abnormal gene, does not appear on the photographic film, correct? So far it is correct. So far, I'm saying so far it is correct. Mutated gene does not appear on the photographic as the probe. Why it is not appearing? Because the probe has no complementarity. An option says probe has complementarity. If the probe is complementary, they both will hybridize, my beta. And if both are hybridized, if the probe is present, probe will give some mark. So mutated gene does not present as the probe has complementarity, no. So this statement is partially correct. Up to here, it is correct. After that it is wrong. Option number 2, mutated gene partially present. No, partially it won't present. Partially because if even a single nucleotide is different, the whole strand will not hybridize. This is wrong. Mutated gene completely and clearly appears. No, we all know that if there are mutations, there are darkness. Okay. Mutated gene does not appear on a photographic film as the probe has no complementarity. No complementarity. Yes, correct. So option number 4 is correct here. As the probe has no complement, then only when the probe cannot hybridize, then only it will be seen. Okay, it won't be seen, and there will be darkness. If the probe is hybridized, probe will certainly present in the sample. At the time of photographic film, the probe will give some radiation because the radiations are coming from within. Therefore, we use the term autoradiography. The mark is due to radiation, and radiations are not used from outside. They are coming from within, auto, self. Therefore, it is used, the term is autoradiography. Make sense? So, yes, it is a good question, but statements are there. And if you don't read the statements, and if you don't understand also, then also, you won't be that much confident in your answer. Erythropoietin hormone, which stimulates RBC formation, is produced by. Now, I remember 10, 12 students after the test, they said, Sir, ye kahan se aa gaya? It is not in the NCRT. It is there in the NCRT. In the chapter of chemical coordination, the last few pages, if you see, hormones from the other part, kidney, GI tract, those hormones, you will find erythropoietin is secreted by the JG cells, juxtaglomerular cell. If you don't learn it, okay, if you know, it's fine. And if you know, tell me which other hormone is secreted by the JG cells. Come on. Which other hormone is secreted by the JG cells? Yes, your answer is correct. 2 answers are correct. Rahul, Priyanka, answer is correct. Mahima, answer is correct. And Beta, Lenovo, your answer is partially correct. Your spelling is wrong. Lenovo K6 Note. Mehroof. Yes, 2000, answer correct. Realme, answer correct. Lenovo, now your answer is correct. Yeah. Now your answer is correct. Yes. To me, answer is correct. Yes. So, answer is renin. And you have to tell me when the JG cell produce renin. When? When the blood pressure is low or when the blood pressure is high? When the BP is low, then renin, or when the BP is high, then renin. Come on, tell me. When they will be producing renin? Come on, tell me. Low, high, low. At least out of 4, 75% of you give the correct answer, and the correct answer is low. When the blood pressure is low, at that time, the JG cell gets stimulated and they produce the hormone renin. Renin is an enzyme, but it works like a hormone. So it is somewhere between enzyme and a hormone. Very good. And the spelling of renin is RENIN. JG cells also produce a hormone erythropoietin. Erythropoietin hormone is against lower partial pressure of oxygen in the blood. Okay, so in the oxygenated blood, what is the normal partial pressure of oxygen? In the oxygenated blood, how much is the partial pressure of oxygen? Come on, partial pressure of oxygen, PO2. How much? Yes, please. I'll take it. No problems. Okay, I'll take it. It is 95 mm of mercury. 95 mm of mercury. PO2. If the PO2 is maintained lower than this, if in the blood the PO2 is not rising above 70 or 75, in that case the JG cells, they produce erythropoietin. And what this hormone erythropoietin does, come on, what is the effect of erythropoietin hormone? I'll give you the term. Erythropoietin help in erythropoiesis. Once again, erythropoietin helps in erythropoiesis. Now you have to tell me, what does erythropoiesis means? What is erythropoiesis? Yes, it is RBC formation. Yes, the stimulation of the red bone marrow. As a result, RBC formation is stimulated. So erythropoiesis. Poiesis, Poiesis. It means RBC formation. As a result of erythropoietin, the RBC count, the total RBC count increase. So if a person has got erythropoietin produced by the kidney, his RBC are more than the RBC of a person where the kidneys are not producing erythropoietin. And this is in response to lower partial pressure of oxygen. You have to tell me in which condition the kidney will produce erythropoietin. Number one, when the person is living on a plane, or when the person is living on the mountain or at an altitude. At an altitude. When, in which condition erythropoietin will be produced? Answer number one, in altitude. Answer number two, only in the plane. Answer number three, that in both. In both the conditions, erythropoietin is produced. And answer number four, in none, in no condition. Okay. Option number one, altitude, more RBCs. Option number two, plain, more RBCs. Option number three, both have more RBCs. In both the condition, erythropoietin produced. Option number four, in none of the condition, erythropoietin produced. Answer is option number one, yes, altitude. Very good. At altitude, the erythropoietin hormone is produced, and as a result, those who are living at high altitude, they have got higher RBC than those who live in the plains. Okay? Now tell me, what is the normal RBC count? Give me the answer of NCERT. I want you to remember, but equal to the answers of NCERT. What is the normal RBC count? Come on. Yes, it is 5 to 5.5 million per millimeter cube, per cubic millimeter. 5 to 5.5 million per cubic millimeter. Very good. All the RBC count, WBC count, any count, platelet count, they are always counted in per cubic millimeter. Remember this, thumb rule. Any count, RBC, WBC, differential WBC, total WBC, or platelet count, it is always in per cubic millimeter. Okay? If someone asks you how much is the blood hemoglobin, give me the concentration of blood hemoglobin. Yes, come on. What is your answer? How much hemoglobin is present in the blood? Come on. All these are questions of your NCERT. I am not giving anything new, beta. All these are NCERT questions I am discussing. Yes, you are right. Read me, your answer is correct. You are giving correct answer, that too very fast. And that is a good sign. Yes, answer is 12. And Mahima also gave the correct answer. Gayatri gave the correct answer. Rajendra gave the Mahima, Priyanka. Yes, very good. In fact, the answer of Priyanka is the best so far, because Priyanka has given the complete answer saying that 12 to 16 gram per 100 ml of blood. Any other concentration is given in per 100 ml of blood. Only the count, the cell count is given in per cubic millimeter. All the other values are given per 100 ml of blood. What is the glucose fasting condition? 60 to 100 gram per 100 ml of blood. You understand? Very good. So 12 to 16 gram is the hemoglobin concentration per 100 ml of blood. Very, very good. Okay. Achha, if you have concentrated in my previous class, I want you to answer a question. If a person has anemia, when the doctor diagnosed, the total hemoglobin is found to be 8 milligram per cubic millimeter. 8 milligram per cubic, sorry, 8 gram, it is a gram percent na, 8 gram per cubic per 100 ml. 8 gram per 100 ml. That is in anemia, this is a amount of hemoglobin. Find out that total oxygen carried by oxygenated blood. Find out the total amount of oxygen carried by oxygenated blood. Come on, let us see. How much? Question says that anemia is present. The total hemoglobin content is 8 gram per 100 ml of blood. Rather than 12 to 16, it is 8. In that